Continue. Hide. Okay, cool. All right, so we're gonna start by talking about incomplete and codominance, and these are basically just genetics things that happen that are different than just dominant or just recessive. When it's just dominant or just recessive, how many different phenotypes or like physical appearances are you gonna see in the population? 2. You're gonna see some things that are dominant and some things that are recessive. And probably whatever's dominant is gonna be more common. That's pretty normal. But it's not always the case. Having 6 fingers is a dominant trait. Having dwarfism is a dominant trait. Let's see what else. Marfan syndrome is a dominant trait. None of you have those things. So that's just to say, like, just because it's dominant doesn't mean that it's necessarily more common, even though usually it is. Okay. With codominance, instead of seeing just 2 options, you're going to see 3 options. So what happens in codominance is 2 traits are both dominant. So what you're going to see, those phenotypes are going to be 3 options where one is a mix of the other 2. Now, when I say a mix, I mean like a mix where you can see both of the other traits individually. So what I mean by that is like a fruit salad where you have a fruit salad of only strawberries and you have a fruit salad of only bananas. And then you also have a fruit salad of strawberries and bananas where you can see the strawberries and you can see the bananas and they're not like all mixed and blendy. You can still see them, okay? So an example of this in real life would be like you have a bunch of cacti. You have some cacti that have 1-pronged spikes. You have some cacti that have 2-pronged spikes like these. And you have some cacti that have a mixture of some 1 spikes and some V spikes. So you see both, okay? When you do this, you're going to use 2 different capital letters. And the reason you're doing that is because they're both dominant traits. So you're going to have no lower cases and you're going to use only caps. And we're going to do an example in just a second with cow. Blood type. And let's look at it now. Okay, so example problem. This is very basic. We have 2 roan cows. Roan means that they have red and white fur. And if you're curious, you can Google a picture of a roan cow, and it's literally a cow that has like brownish red, like rust colored hairs and also white hairs. It has both the hairs. It doesn't have pink hair. It doesn't have tan hair. It has both hairs at the same time, okay? They're going to mate. Is it possible for them to have a red-haired calf? So the first thing that you should do when you do a genetics problem like this is you should decide what letters you're going to use. Like I said before, it truly doesn't matter what letters you want to use, but I like to use whatever makes sense. So I'm going to use R to stand for red and W to stand for white. Now, the most important thing about this is that you do not set a letter to equal roan, because why can't a letter equal roan? Yeah, because it's a mixture. So you can't have one thing that equals a mixture. That doesn't make sense. So now we're going to break this down like really simple. You don't have to do this when you solve these problems, but for this one, I think it helps. So we're going to do possible genotypes are 2 capital R's, a capital R and a capital W, or 2 capital W's. Those are all the possible combinations of R's and W's that you could have. And then we'll say like, what are the phenotypes going to be for those specific cows? So if a cow has 2 capital R's, what color will they be? Red. If they have a capital R and a capital W, what color will they be? Roan or mixed. And if they have two capital W's, they will be white, plain white. And then just to like reinforce this, I'm going to do the like term that describes the genotype here. So two capital letters that are the same, what do we call that? Do you know? Starts with a homozygous. And I'm just going to write a D for dominant, but that's what it is, homozygous. Two of the same is homozygous. What is two different? Good job. Heterozygous. And then two capital letters, this is also homozygous dominant. If you had two lowercase letters, it would be homozygous recessive. Two of the same and small would be recessive. Okay, now the next thing that you need to do is you need to figure out what are the parents' genotypes. And to do that, you always go back to the question to find what are the parents' genotypes. So this one said two roan cows are the parents. So that's easy because we've got our mom crossed with our dad, and they're both going to be the same. They're both going to be RW because they're both roan. And then you can do your square. So draw yourself your little box. You only need 4. And put your eggs, possible eggs going in one direction. Doesn't matter if they're on the top or the side. And your possible sperm going in the other direction. And then fill her in. So RR, RW, RW, WW. And just like I said with the capital letters, always write either your R's or your W's first. So since I started over here with writing my R's first, I'm going to stick with writing my R's first so that it's easy for me to figure out that these two things are the same as each other. Because if I wrote RW here and WR here, I might be more likely to get confused, especially if I was rushing. So write them always the same. Okay, and then our question, which is what the whole point was, is it possible for them to have a red-haired calf? Yeah, yes. And that would be all you'd have to write for this question because it didn't even ask you about a percent. If it did ask you about a percent, it would be 25% chance because only this one is red. Huzzah. Okay, now a little more complicated example of codominance is blood type. So we're going to talk about that. I put it over here to the side if you're using OneNote. Okay, so this shows the possible blood types for humans, not considering positive or negative, which the positive or negatives, if you know your blood type, you might be like, I'm A positive. Positive is a different gene than AB or O or AB. Okay, so I'm only looking at A's and B's right now. So this is codominance because you have A, which is dominant, B, which is dominant, and O, which is recessive. So when you look at this, don't ask me why I use I's because honestly, I have no idea. And little I. So this is type A. This is type B. This is type O. Why is there not an allele that stands for AB? Like, why did I not put AB up there right now? Yeah, because they're a mixture. It's just like with the roan cows. Like you don't have a letter stand for roan. You also don't have a letter stand for AB because these are not genotypes, they're alleles. Alleles are one letter. So with genotypes, which is what it has here, then you can start to get ABs because you're having one letter that comes from the mom and one letter that comes from the dad. When you put them together, you get the full genotype. I'm just going to make a note here that says genotypes are 2 letters. 2 letters. And the only time that things get a little bit crazy with blood type is noticing the A's and the B's. If you're type A or you're type B, you have an option where you could be homozygous dominant. That means I big A, or you could be I big A, lowercase I, heterozygous. And either way, you would still have type A. And same thing with B. You could be homozygous or you could be heterozygous, and either way, you still have type B. Because what happens to the little I? Yeah, it's covered up, so you can't see it. If you have both an A and a B, though, you're going to have both traits. And if you have two little I's, you're going to have O. And what this is actually coding for, if you look at the pictures, is the glycoproteins that stick off of your red blood cells. And you have specific ones based on which blood type you actually have. So this is why if you're a type A person, you can't get a transfusion of type B blood because your immune system, like we talked about before break, will recognize that these glycoproteins are foreign to you. And they'll freak out and you'll go into septic shock and you could die from that, from getting the wrong blood transfusion. So super bad. This is also why type O can donate to anyone because their blood cells are like stealth mode. Like they don't have anything on them. So any person can get these and their immune system won't recognize it as being foreign, which is why if you are type O and you donate blood, or your parents are type O and they donate blood, like the Red Cross will call them or you and be like, hey, we really want will you please donate? Because there actually is a blood shortage right now. I don't know if you know that. So if you're considering giving blood, it's a blood drive, then maybe go for it because there is actually a shortage of blood donations, like nationwide. Anyways, and last thing about this is that people who are AB, they can receive blood from anyone because they recognize the A glycoproteins as supposed to be there. They recognize the B glycoproteins as supposed to be there, and O they can receive, of course. So I'll just make a note that says O is universal donor, AB is universal recipient. And to be more specific, O negative is the universal donor because they also don't have RH, which is the other like positive or negative. One sec, I'll get you. And AB positive is the universal recipient because they have everything. So basically, O negative people have nothing. So like super stealthy. And AB positive people have everything. So they won't recognize anything as bad. Go ahead, Savannah. Because if AB gives to B, then the A's that are on the AB will get recognized by the B person's immune system and like freak out. And same thing with this, and same thing with this. Yeah. Yeah, people with O blood can only receive O blood.
How frequent, like what percentage of people?
I think it's fairly common. Can someone look up most common blood type? I know AB is the least common. But you guys need to see a Punnett square with this stuff, or do you feel okay about it? See one? You're good? Okay.
Can you show one?
Yeah. Oh, negative is 7%?
Yeah, O positive is 38.
So I think O in general is the most common and AB is the least common. Okay, let's do a like little fake Punnett square here. So we're gonna say, I'm just making this up. A man with say, A blood whose father has O blood, and a woman with AB blood have a baby. What are the possible blood types? Okay. All right, so when you do this, you're going to look at first of all, the man. The man has A blood. So you know, I'm going to do my little lines, dad and mom. So you know that dad has to have at least one I, capital A, because he has A blood. And his father had O blood. This is kind of like a check-in. What would be the other letter? Yeah, little I. You don't have to write O with a little I because it's not capital, it's the only little I. And the woman has AB blood. So she's I, capital A, I, capital B. And then when you do it, just make sure your superscript letters like travel with you. Don't lose them. So then you have I, capital A, I, capital A, I, capital A, small I, capital A, I, capital B. Always write your A's first. I, capital B, small I. So then my answers would be, I didn't ask for percents, but I'm going to put them anyways. This would be 50% type A because both of these are type A. And then 25% type AB and 25% type B. Now, I do want to tell you that if you hate the I's and the superscript letters, you can do it. I'm going to put this over here. Or you can do this if you like this better, but you have to remember that this is recessive. So when you do this, you would have this. And you need to remember that these would just be A and this would just be B. The O is like silent unless it's OO. So if you are like looking at this with the eyes and you're like, I hate this with my whole heart, and this makes you feel better, fine, go for it. But just don't forget that this is not type AO. It's just A. Okay. Okay, that's blood type. Let's talk about one more type, which is incomplete dominance. And this is similar to codominance, but different. So this is going to be where one allele is partially dominant, but not all the way. So when you see incomplete dominance, you're going to see, again, 3 phenotypes, 3 physical appearances, where one is a blend of the other 2. So if before we were talking about a fruit salad where you had the fruits and you could still see the strawberries and you could still see the bananas, now I'm talking about a fruit smoothie where you can no longer see the strawberries and the bananas. You see a pink blend of both. Okay, that's what this is. So some examples in real life are things like straight hair, wavy hair, and curly hair, right? Or like brown, tan, white, where what I've written in the middle is the blend or the mixture, okay? When you do these problems, you use what we call a prime. So use one capital letter and the same capital letter quote prime is what it's called. And what this looks like is R and same capital R with an apostrophe after it. That's the prime. This thing, prime. And again, I don't know why it's called that, but it is. We'll do our little sample of this. And typically, like when I was writing this, I wrote it pretty fast, so I didn't give like a lot of background info. But if you saw a problem like this, it would probably tell you like, there's a field of red, white, and pink flowers. So you would know, okay, there's 3 things and one of them's in between the other. When I wrote this problem, I didn't write that. I just wrote, there's a red and pink flower because I put it in the incomplete dominant section. So like, obviously it's incomplete dominant. Okay. So first of all, you're going to decide your letters. Again, I'm going to use the letter that just makes sense for the problem. So I'm going to use R. So capital R and capital R prime. And capital R, I'm going to make it stand for red, and capital R prime, I'm going to make it stand for white and not pink because pink is the mixture. Okay. One thing about this is it literally doesn't matter if you want to flip it. If it was W for white and W prime for red, it's fine. It doesn't matter. As long as you choose the ones that are on the edges and not the mixture thing. Okay. So this is my little like chart where it's 2 capital R's, capital R prime. I always write my prime second because if you write it in the middle, it's easy to lose it and forget that it's there. And then this one's R prime, R prime. These are, what is the word to describe this group of letters? Genotype, yes. Okay. And now we're going to think about what do these look like. So what they look like if they have red plus red, they're red. If they have red plus white, they're pink. If they have white plus white, they're white. Are you guys having fun? What is the word to describe these? Yeah, phenotype. Okay, and then again, these are homozygous. I'm gonna write homozygous dominant here. And again, remember, like, but star, not all the way. And then this bad boy in the middle is still heterozygous. Okay, and then to get your parents, you need to go back to the problem. So red flower and pink flower. 2 R's, R prime, fill her in, R prime, R. It's like therapy, guys. It's so nice. Yeah. So like either of them could be not all the way, and it doesn't matter which. And that's why I started both, because no matter which one it is, you're going to get the same result. All right. And then the question said, what percent of the offspring are red? 50. So summary of that is codominance, you're going to see both things, but separately. Incomplete dominance, you're going to see a mixture or blend of the things. Okay, fruit salad, fruit smoothie. Good. All right, before we go on from genetics, I want to tell you about some weird things that happen that are like, what's going on? So if you're on OneNote, look at this thing that says weird stuff. And we're gonna start with this. That's my cat, that's soybean. Okay, so one thing that's weird is we talked about how females have 2 X chromosomes. X chromosomes have a lot of information included on them. So much information that if you are a female, one of your X chromosomes is actually inactivated in every single cell that you have. And for humans, it doesn't really matter because there's no traits on the X chromosome that are like outwardly physically manifesting, except for like lymph node patterns, which they're really not outwardly physically manifesting. But in cats and horses, the fur color gene is on the X chromosome. And so what happens is it's just like random which X is activated and which X is inactivated in each cell. So that's why in female cats, you can get this, like Soybean. She's a beautiful tortoiseshell cat, and she has like little milk mustache of like lighter fur and then like brown fur in other places. And same with calico cats, where they have like orange, white, like brown or black. That is because of X inactivation, where like they're heterozygous. So one of their X's is activated, the other one's turned off, in a different part of their body, it's switched, which is like very cool. And this is also why in male cats, you're going to get male cats that are either black or orange, but you're going to get female cats that are calico or mixed or blended. So that's an example of this. It's actually like not an incomplete dominance or codominant situation. Like if you look at this, you would be like, wow, codominance. It's like both are showing fruit salad, but actually it's not. It's just that half of the genes are turned off in those specific cells. So that's what's happening with this. The inactivated X is called a Barr body, which is this right here. It's just named after the scientist who discovered them. And then also a weird thing is that sometimes genes are linked if they're found on the same chromosome. So remember when we talked about meiosis, how we talked about like the independent assortment where like they separate, but if they're on the same chromosome, they don't separate as often. So sometimes you're going to see like two traits that are almost always inherited together. The only way to get them apart is through crossing over, which happens sometimes. But if they're really close together on the chromosome, then are they likely to be split up by crossing over? No, because it's like trying to, like, let's say they're here and here, you would have to cross over exactly at this spot to get them separated. So I'm just going to make a note that says, if genes are close together on chromosome, less likely to be isolated, separated by crossing over, which equals linked. And then here's just a picture of a cat that's not as pretty as Soybean with inactivation, kind of showing you like what's going on there. And they literally are like the inactive X's are like scrunched up. It's like they're like putting you in a box. Goodbye, which is kind of interesting. But this is X inactivation. Okay, more weird things. Things like eye color. So you are, I'm sure, aware that there's a range of eye colors. It's not just like 2 options or even 3 options. There's a lot more than that. And that's because there's more than one gene that determines eye color. So to have blue eyes, you have to be recessive on both the genes that control eye color. If you're recessive on only one of them, you're going to have something in the green-brown range. And if you're recessive or dominant on all of them, you're going to have brown eyes. So this is called polygenic traits, which means more than one gene controls them. So you're going to get a range of phenotypes. Can you think of another example of this in humans? Where you see a range of things? No? Yeah. Hair color, skin color, height. Things where you see like if it wasn't polygenic and we're talking about height, it would be like you're like 5'5" and you're like 6'7", and that's what it is. And everyone's 5'5" or 6'7", except for environmental factors, and like that's it. Like no, there's many genes that are affecting that, plus environmental factors. So we get a large range. Okay. Okay, that's polygenic traits. Now let's talk about the environment. So environment can also impact gene expression. And in humans, these are things like nutrition, environment, like those sorts of things can affect whether your genes are like actually determining your height or whatever. Like environmental factors are important. This example is with hydrangeas. And do you know about hydrangeas, like why they are different colors? Why? I don't know. Yeah, the pH of the soil. So this is an example. Hydrangeas are different colors based on soil pH. So if you ever go to the beach, you will see, and you have hydrangeas. Like my mom has hydrangeas, they're blue like this. But if we go to the beach, the hydrangeas are more like purpley or like even pink. And it's related to like the soil and not related to the traits that they have, which is just like kind of cool. So environment can cause different genes to turn on or off. It's not always just like you get what you get. Kind of cool. Okay. Then there's also this that we have to talk about. And this is the last weird thing. And then I'm going to talk about pedigrees. So weird things. Different traits can look like normal dominance, incomplete dominance, and / or codominance if you look at them on different scales. So when we're talking about Tay-Sachs, you remember Tay-Sachs? It's a genetic disorder where people can't break down lipids, and it's actually very sad because usually people who have Tay-Sachs don't live past like 3. There's actually a Grey's Anatomy episode from like many seasons ago about Tay-Sachs. It's very sad, but if you watch it, you can learn more about it. So when you look at a person, a whole person, it looks recessive because to have Tay-Sachs, those children have 2 recessive alleles. So they have 2 small letters. But if you look at heterozygous people and you look at their like biochemical composition, like what's actually going on, it looks like incomplete dominance when you look at that, if you took like a blood test, because they have somewhere in the middle in terms of their ability to metabolize lipids. It's not so bad that they have Tay-Sachs, but it's not as functional as a normal person. So it looks like they're in the middle, incomplete dominance, blendy. If you look at their molecules, the heterozygous people, they're producing half functional enzymes and half non-functional enzymes. So what it looks like then, if you look at the molecules, is you look at codominance. Because you're like, oh, it's a mixture, like half are this and half are that, so codominance. So this is just to say, like, genetics is not as clear cut as probably you learned freshman year, where it was like, this is this, hooray, done. It's actually a lot more complex about it, and scientists are still really learning what's going on in a lot of ways. We're also not even going to talk about epigenetics, which, have you guys heard of that? So epigenetics it means like above the genome, and it's the study of like how the environment is turning on or off different genes, and how your genes being turned off, you actually could pass on to your children. So there's studies about like people whose grandparents lived through a famine and they have different genes activated versus people who have grandparents who didn't live through a famine. So it's like very interesting that like what you do in your life and what happens to you in your life can actually affect your DNA, and that you can actually pass that to your children and your children's children, which is nuts. So that's epigenetics. It's still like very new. Okay, let's talk about pedigrees real fast. So pedigrees are these thingers. And they are basically just family trees that show the passage of one specific trait. So pedigree equals family tree shows passage of one trait. And if they have the trait on the family tree, they are colored in. So colored in equals has trait. If they're square, guess what are they? There's only two choices. Male, good guess. If they're a circle, they're a female. If they're slashed out, they die. Okay. This is our little like key. If there's a horizontal line, that means they're married. So this square is married to this circle or was. If there's a line going down, that means that they are children. So this square and this circle had 1, 2, 3, 4 children. They didn't have 8 children because only the people connected by these down lines are actually their children. Okay, the other ones are married in. So what's cool about pedigrees is that you can actually use them to figure out whether a trait is dominant, recessive, sex-linked, or autosomal, which autosomal just means not sex-linked, not linked to the X chromosome. So over here, I'm going to make us a little guide. Won't let me though. Okay, so if trait is dominant, what you're going to see is that you could have 2 parents with the trait who have an unaffected child. And what that would look like is square, married to circle. Both of them are colored in because they both have the trait. Their child is not colored in. And the reason that you could have that is we've got our square here. I'm going to use R's. So this square would be capital R, lowercase r. This circle would be capital R, lowercase r. And then what happened is both of them passed their lowercase r to their son in this case. And that's why he doesn't have the trait. Make sense? If the trait is recessive, if trait is recessive, then you can have 2 unaffected parents who have an affected child. And that's going to look like square, married to circle, child, colored in. Now that we know the trait is recessive, our child is 2 little r's, which means both parents had to be what? Same as the other side. And in this case, there's a word that we would call these parents. You know what it is? Yeah, they're carriers. So these are carriers. If a trait is dominant, can you have carriers? No, because anyone who has a capital R is going to be colored in. So the carriers will just like look like they have the trait. If you have a recessive trait, you can have carriers. Now let's talk about sex-linked versus autosomal. So if the trait is sex-linked, who do you think has it more often? Males or females? It's men, males. More males. Shaded in. And that's because males only have 1 X chromosome. So they don't have any like backup if it's a recessive trait that would cover it up. Like if they get 1 copy, they have it. Females, they have to get 2 copies to have it if it's recessive, which usually if it's X-linked, it is usually recessive. And then if it's autosomal, which just means it's on chromosome 1 to 22, it's not on the 23rd chromosome, then equal-ish numbers, male, female. Now, because remember, like this is probability, so like the chances that you have exactly perfect 50-50 males and females is pretty slim, but it should be like a lot closer than if it's sex linked. So let's try one. Look at this one. And what do you think? Sex linked or autosomal? Who's colored in more? Males. So do you think it's sex linked? Yeah. So this is X or sex linked, either one. And do you think dominant or do you think recessive? Recessive, is right. So now let's just find like a couple of these people's genotypes because that's what is solving a pedigree, you find their genotypes. So when you do this for sex-linked traits, it's a lot easier if you start with the males because they only have one X. So all the males, I'm going to go ahead and fill them in with XY because that's what they have. And then for the ones who have it, will their X have a capital letter or lowercase letter? The trait was recessive. Lowercase. So I'm going to use R's again. So all the males who are shaded in, they have to have a little r. And all of the males who are not shaded in, what do they have? Capitals. Oh, and this guy. Okay. Now you can work backwards and solve the females. And the females have to have 2 X's. So let's look at this woman here. She doesn't have the trait, so she has to have at least 1 capital letter, right? And then what about her second letter? What do you think? Big or small? This. Small. Let's see how we got that. So to figure it out, you look at her sons, because her sons, to be sons, they had to get the Y chromosome from their dad, which means if you're a boy, you got your X chromosome from your mom. Whatever's on it, that's what you got. So this son here had to get this X small r from his mom because he got his Y from his dad. Okay, so she has to be a carrier. Now let's look at this woman. What would she be? Is she a carrier? Yeah. She's a carrier because her son has it. Okay. What about this woman? Can we know? We don't know. So if you don't know, you write what you do know, which you know she's at least 1 capital R because she doesn't have it, but question mark because you don't know what the other one is. Because her daughter down here, we know that her daughter is a carrier because why? Yeah, because she's a girl, so she had to get her X from her dad. So she has to have a small r, but she also has to have a big R because she's not colored in. Okay, are you guys getting it? Okay, let's look at this one. First of all, sex-linked or autosomal. Gosh, you guys, it's rough today. What do you think? Is it different than this? Yeah, this one is not sex-linked, it's autosomal. So this is autosomal. And then do you think dominant or recessive? Look at this family, this part of the family. Slash, just guess if you're not sure. Wow, recessive, because if you fill this in, this one is, I'll use different color. This one is too small letters, recessive. These people had to be carriers. Okay. That's why all these people are like this, because there's no dominant letters in this side of the family. Everyone's lowercase. Lowercase, capital lowercase, lowercase, lowercase. These people, do you know what they are? Capital R question mark. Because they could be carriers, but they could also be normal, just like regular, two capitals. Okay, that was fun. Any questions? All right. What we're doing now is stopping this.